Cloning. I mean, just say the word, right? It immediately makes you think of stuff straight out of science fiction. You're probably picturing armies of identical soldiers, maybe bringing back dinosaurs, or, the classic evil twin. But the real story of cloning, it's way more subtle, and honestly, a whole lot more fascinating than what you see in the movies. So let's get into it. Let's separate the science from the sci-fi.
So yeah, when I say cloning, what's the first thing that pops into your head? For most people, it's all that stuff from books and movies. But here's the thing. Cloning isn't just one single thing. It's more like a whole collection of different tools. And to really get what it's all about, we've got to look past all that Hollywood stuff.
And what's really wild is that nature, it's been cloning things for billions of years. Seriously, from tiny bacteria just splitting in two, all the way up to these massive aspen groves, where every tree is actually part of the same single organism. But the best example, the one we can all picture, identical twins. Yep, they are 100% natural clones. It happens when a single fertilized egg just splits, creating two people with pretty much the exact same DNA.
So when you boil it all down, that's what cloning is at its very core. It's just the process of making genetically identical copies. And that copy could be a whole organism, a single cell, or even just a little tiny piece of DNA. Okay, that's our foundation. Let's build on that.
All right, here's a little fun fact for you. The word clone doesn't come from some high-tech lab of the future. Nope, it comes from ancient Greece. The original word, clone, literally means twig. And that just perfectly captures the original idea, right? You take a twig, a little cutting, from a plant, and you grow a whole new, identical plant from it. I mean, the idea is as old as gardening itself.
Okay, so when we take this idea into a modern lab, scientists are usually heading down one of three main paths, and each path has a totally different end goal. We're going to break down reproductive cloning, therapeutic cloning, and molecular cloning. Let's kick things off with the one you've definitely heard about, the one that gets all the media attention.
Right. Reproductive cloning. This is the one that makes the headlines, the one that starts all the big debates. We're talking about making a whole, living, breathing copy of an animal that already exists.
The goal here is crystal clear. Make a brand new individual that's a genetic twin of the original. You can think of it as a kind of artificial version of asexual reproduction, making a baby from just one parent. And the main technique they use to pull this off has a fancy name: somatic cell and nuclear transfer.
So how in the world does that actually work? Well, it's kind of like a transplant, but on a totally microscopic level. First, you take what's called a somatic cell. That's just a fancy term for any regular body cell, like a skin cell from the animal you want to clone. That cell has all the DNA, the complete instruction manual. Then you get an egg cell from a donor, and you carefully scoop out its nucleus, basically erasing its original genetic info. Now here's the magic trick. You take the nucleus from the skin cell and pop it into that empty egg. You give it a little zap of electricity to wake it up, and suddenly the egg thinks it's been fertilized. It starts dividing and becomes an embryo that's a perfect genetic match to the original animal. And from that point, it's implanted into a surrogate mom to grow just like any other pregnancy.
And this exact process is how we got the most famous sheep in history, Dolly. She was born back in 1996, and she was a massive deal. I mean, the first mammal ever to be cloned from an adult cell. That had never been done before. When they announced her to the world a year later, it was a media firestorm. It totally changed how everyone talked about cloning. And she lived a pretty normal sheep life. She even had her own babies before she passed away from a lung disease that's actually pretty common for sheep.
But man, making her was not easy. It took the scientists 277 tries, 277 attempts to finally get the one successful embryo that grew into Dolly. Just let that sink in. That's a success rate of what, less than 0.5%? It just goes to show you what an incredible challenge this was, and how many, many times it didn't work before they had that one huge success.
Okay, let's switch gears a bit and talk about therapeutic cloning. Now this is really interesting because it starts with the exact same technique we just talked about, but the goal is completely different. We're not trying to make a whole new person or animal, we're trying to create new hope for medicine.
So here's the key difference, the real fork in the road. Both types of cloning start by creating that cloned embryo. But with reproductive cloning, you implant it in a uterus to grow. With therapeutic cloning, you stop. The process ends right there. That embryo is never, ever implanted. Instead, the whole point is to harvest its stem cells, these amazing, super versatile cells, and use them in a lab.
So why would we do that? Well, the potential is just enormous. Seriously, just imagine. Imagine being able to grow new tissues for a patient that are a perfect genetic match, so their body would never reject them.
We could, in theory, grow new heart muscle cells for someone who's had a heart attack, or create new insulin-making cells to treat diabetes, or maybe even grow nerve cells to help fix a spinal cord injury. This is the stuff of regenerative medicine, and it could be a total game changer.
But of course, this incredible promise comes with a massive ethical question mark. To get those stem cells, you have to create a human embryo, and then you have to destroy it. And this has sparked a really intense, really difficult debate. When does life actually begin? Does an embryo that was only ever made for research have the same moral status as one that could become a person? This is the issue that's right at the very center of the whole therapeutic cloning debate.
All right, on to our third and final type. And here's the interesting part. This is actually the most common, most widely used kind of cloning out there, even though it's probably the one you hear the least about. We're talking about molecular cloning, and it is an absolute workhorse of modern biology.
So the big difference here is all about scale. We're not talking about copying a whole animal anymore. We're talking about copying tiny, tiny little segments of DNA. So think about it this way. It's less like cloning a whole sheep and more like using a photocopier to make millions of copies of just one single page from a giant encyclopedia. Scientists find the specific gene they want, they stick it into a little carrier, usually a bit of bacterial DNA called a plasmid, and then that bacteria does all the work, churning out millions of perfect copies of that gene.
And this technique, it is everywhere. This is how we make huge amounts of life-saving medicines like insulin. Before this, insulin had to be taken from animals. This is a core part of how we develop new vaccines, how we create tests to screen for genetic diseases, and yeah, it's used in agriculture to create genetically modified organisms, or GMOs. It is a fundamental tool that has absolutely revolutionized medicine and biotechnology.
So let's just pull this all together for a second. You've got reproductive cloning, which is all about copying a whole animal and brings up these big questions about what it means to be an individual. Then you have therapeutic cloning, which is about making stem cells for medicine, and that conversation is all about the ethics of using embryos. And finally, molecular cloning, which is just about copying bits of DNA, a standard tool that leads to discussions about things like GMOs. See, 3 totally different tools leading to 3 totally different, really important conversations.
So if there's one thing you take away from all this, let it be this. Cloning isn't just one thing. It's not a single concept. It's a name we give to a whole toolbox full of powerful biological techniques. And each tool in that box has a very different purpose, a different potential, and brings up its own unique set of ethical questions. And just knowing the difference between them, that's the absolute first step to having a smart, meaningful conversation about any of it. Look, the potential here is it's undeniable. We're talking about everything from maybe saving endangered species to curing horrible diseases. But these tools also give us this incredible, unprecedented power to rewrite the building blocks of life itself. So the real question at the end of the day isn't just can we do this, it's should we? And maybe even more importantly, how do we, all of us as a society, decide where we draw the lines? That's a question that science on its own just can't answer.